Hi, and welcome back to Chapter 12, Part 3. In this lecture, we're going to explore how molecular tools like electrophoresis, PCR, and sequencing allow scientists to analyze genes and proteins. Before jumping onto the molecular analysis of proteins, though, let's do a quick recap of our previous video. So the first checkpoint question asks, what does a DNA probe consist of? Remember, a DNA probe is a short, single-stranded DNA fragment with a complementary sequence to some kind of target DNA region. And these probes are labeled with either radioactive or fluorescent tags so that they can be visually detected when they bind or hybridize to their matching DNA. And our second checkpoint question asks, why is a southern blot used after gel electrophoresis of a DNA digest? So after gel electrophoresis, DNA fragments are separated by size, but remain mixed in the gel. So the southern blot can transfer these fragments to a membrane. So here's my membrane, which can then be probed by using a labeled DNA sequence. So the probes we just talked about up here. So this step identifies whether the specific target gene or sequence is present amongst all of those fragments. DNA and RNA tell us what could happen in a cell, but proteins reveal what's actually happening. So to study active gene expression or disease states, we often need to examine proteins directly. So we'll start by looking at polyacrylamide gel electrophoresis. So protein analysis helps us identify which genes are active and how cells are responding to their environment. This is really key for diagnosing diseases and identifying microorganisms. And the most common method is polyacrylamide gel electrophoresis, abbreviated PAGE, which separates proteins by size. In SDS PAGE, the detergent SDS, which stands for sodium dodecyl sulfate, unfolds proteins and coats them with a uniform negative charge. And then when we apply an electrical current, smaller proteins will travel faster through the gel. Staining with Kumasi blue or silver stain reveals distinct protein bands, and that allows us to estimate size and abundance of the protein. Here you can see the SDS PAGE setup and results. So up here we can see that SDS is a detergent that denatures proteins and masks their native charges, making them negatively charged uniformly. And then by the end of the electrophoresis, we can see bands of proteins, and each band represents proteins of different sizes that have migrated based on molecular weight. And we will always have some kind of standard, just like we did for our DNA gel electrophoresis in agarose, to act like a ruler to estimate protein sizes. This question asks, how are proteins separated by SDS PAGE? So SDS PAGE stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis, and that separates proteins by size, or molecular weight, you could say. And the SDS detergent, we saw denatures proteins, giving them a uniform negative charge, so migration or movement through the gel only depends on length. Smaller proteins will move faster through those gel pores. Larger proteins will stay towards the top. All right, next we're going to move into amplification-based DNA methods. These include PCR, polymerase chain reaction, and DNA sequence. And these allow us to detect, copy, and read genetic information in precise ways. Polymerase chain reaction, or PCR, amplifies a specific DNA target millions of times in a test tube, and it relies on an enzyme called Taq polymerase. This is a heat-stable enzyme that we derived from the bacterium Thermus aquaticus that was isolated from a hot spring in Yellowstone National Park. We'll also need DNA primers that bind to the target DNA sequences, and repeated cycles of heating and cooling, I'll talk about the specific steps shortly, will produce tons of identical DNA copies within a few hours. So some use cases, just some examples of how we use PCR include for disease detection, forensics, or paternity testing. There are three main steps to PCR. One is denaturation at 95 degrees Celsius, which separates the strands, and this is very hot, and that's why we need that temperature, the heat-resistant Taq polymerase. Here, annealing is our second step, a little bit cooler at 50 degrees Celsius. This allows DNA primers to bind their target sequences. And then we warm it up again with extension at 72 degrees Celsius, which lets Taq DNA polymerase add nucleotides. And repeating these steps over and over again, around 25 to 40 times, leads to an exponential increase in DNA, and this is managed by a thermal cycler machine. And our book gives us a little link to a short animation that's helpful to view the process of PCR. Here we can see what's happening inside the thermocycler. I have denaturation of my original target DNA. I can see they're now single-stranded. In annealing, the primers bind. And then in extension, Taq DNA polymerase is going to copy the rest of the strand. At the end, I have 2 copies of my DNA. And cycle 2, same thing is happening. It looks like they have already denatured. Here's the first strand. Here's that second strand where this is 1 and this is 2. 1, 2. So they've denatured both. The primers are already bind here and here. And then Taq DNA polymerase has already extended the 4 DNA molecules. At the end, I have these 4 complete molecules. At the end of cycle 3, I have 8, and then end of cycle 4, I'll have 16. So it looks like kind of like my generation time calculation from one of our previous lectures. And the same thing would be true where you have your initial number of DNA molecules. So here I only had 1. And the small n over here represents how many cycles of PCR you're going through. So if I started with 1 molecule of DNA at the beginning, and I go through, let's say, 4 cycles, 2 to the 4th, so 1 times 2 to the 4th power, 2 times 2 times 2 times 2, that's 4, so 4 times 4, 16, and I only had 1 to start, so at the end, that's why I have 16 here. So the equation is very similar to the growth we've seen before in bacteria, but now we're looking at doubling of DNA using PCR. And PCR has really revolutionized microbiology because imagine if you're at a crime scene and you see a little bit of blood, maybe there's a little bit of DNA in there to analyze, but it's usually not enough. Luckily, we have PCR. We can take millions of copies just in a few hours in order to do all the tests that you would want to on that DNA. It can identify genetic mutations, forensics, like I mentioned, paternity testing, studying ancient DNA samples. So it's really the foundation for many of the current tests we have today. We also have several variations in PCR. We have RT-PCR, which stands for reverse transcriptase polymerase chain reaction. This adds the enzyme reverse transcriptase to convert messenger RNA into complementary DNA. Remember, that is DNA without introns, and this allows us to measure gene expression. We also have qPCR, also known as quantitative PCR, also sometimes called real-time PCR. This tracks amplification in real time in order to measure the amount of DNA we have. Often this uses fluorescent dyes. And then we have qRT-PCR, so this is combining both at the same time. We're using real-time PCR along with RT-PCR. So Q, which is quantitative, RT, which is reverse transcriptase PCR, is commonly used to monitor viral load in patients with HIV. So if you're looking at therapy, HIV treatment, you can see if the number of viral particles in the patient is going down over time. Now let's switch gears and look at DNA sequencing. So the purpose of DNA sequencing is to reveal the exact order of nucleotides. And a classic method is the Sanger method. This uses chain-terminating nucleotides, also known as dideoxynucleotides or ddNTPs, that stop DNA synthesis at specific bases. Remember that DNA can only grow from the 5' to 3' direction because we need, here's the normal nucleotide, these carbons were, remember they were numbered. This was carbon 1, that was 1', 2, 3, 4, and 5. The 3' end with that hydroxyl group is needed for the next nucleotide to come and bind to extend the nucleotide. In dideoxynucleotides here, there is no hydroxyl group. That means if it were added over here, for instance, and there's no hydroxyl group here at that 3' carbon, the next nucleotide could not be added. So this stops the growing chain. That's why we call it chain termination. And this will produce a series of DNA fragments that can be separated by gel electrophoresis and then read through fluorescent dyes or other kind of some kind of imaging, and we can read this to determine the sequence of nucleotides. So here's an image of that Sanger sequencing. You would need your template strand. Let's say this is my template strand, and let's say my template is this random sequence. Let's see. And then I would need a bunch of regular nucleotides that are not labeled, not dideoxy. So I would just read my template strand, and I would put down, if I read C, I put down G. If I read T, I put down A, I put down T. But then I've also added a bunch of these fluorescently labeled dideoxynucleotides. Remember, these result in chain termination, and you have each dideoxynucleotide labeled with a different fluorescent color. So T's thymines look like red, guanines are black in this case, adenine green, cytosine blue. So here, let's say I read A, instead of putting down a regular nucleotide, I accidentally put down I incorporate a dideoxynucleotide, so the one here. That means I can no longer grow, there's no 3' end, so my chain will terminate here and I'll generate a short fragment. Imagine this is happening thousands of times in the machine, and every once in a while, they're going to randomly incorporate one of these other dideoxynucleotides. So you're going to generate a bunch of different fragment lengths, and we are going to have a detector that can read the different lengths in order and tell us what color appears each time, and we can put all of this information together to generate the actual order of the nucleotides in my strand of DNA. We also have more modern capillary electrophoresis systems that separate fragments by size and read fluorescent signals automatically. So this can really produce high resolution sequence data. So how it works very generally is we're going to run PCR. We have our DNA template, primers, our DNA polymerase, our regular nucleotides, and then we have our fluorescently labeled dideoxynucleotides that cause chain termination. So remember that anytime a dideoxynucleotide is incorporated instead of a regular nucleotide, elongation will stop. This will result in fragments of different sizes, and those different sizes each will have a different fluorescently labeled nucleotide. We're going to run those fragments through a tiny capillary gel, and then our detector can read the different colors that show up, and the computer can identify the sequence for us. So those sequencing methods are good for low volume or small volume cases, if you're just starting to sequence maybe a plasmid. But if you need to handle a large volume of information in a short period of time, we're going to require something that's more high throughput and automated. So that takes us to next generation sequencing. In next generation sequencing, or NGS, this is a really powerful version that can read entire genomes, like the human genome, in a single run. It's faster, cheaper, and it's commonly used in clinical microbiology for pathogen detection and genome comparison. And this really allows us also to store a lot of genetic information and share it in databases like GenBank. So GenBank is a genetic sequence database that is publicly available, and it's at the NCBI, or National Center for Biotechnology Information. It allows us to compare what we get from our NGS results to what we already know, what's already out there. So our book talks about 454 sequencing, or pyro sequencing, but that's really now obsolete. It's been replaced by really these two companies, Illumina sequencing, which is shown on the left, and Oxford Nanopore, which is shown on the bottom right. So in Illumina, we are going to fragment DNA, and sequencing is completed through using uniquely fluorescently labeled bases, similar to what we saw in sequencing for Sanger sequencing, and then we reconstruct the sequence. So what they're doing is taking many, many copies of the genome, fragmenting them, so some parts will overlap with one another, and doing the same basic methods as Sanger sequencing, but large scale. In the Oxford Nanopore, this is kind of cool. We're going to measure current change as the nucleic acid passes through a tiny nanopore membrane that has a voltage applied. And each base, as it passes through the membrane, makes a unique change in the ionic current, and we can get that readout to determine the order of bases in that DNA. Let's do a quick summary of our key tools. And we had DNA probes and blotting that can detect specific genetic sequences. We had electrophoresis that could separate molecules by size. We could separate DNA, RNA, or even proteins by size. Microarrays and PCR can look at differences in gene expression. PCR also helps us amplify DNA, and sequencing helps us read the full genetic code, the order of nucleotides. So altogether, these tools really power molecular diagnostics and the field of genetic engineering. This checkpoint asks how PCR is similar to natural DNA replication versus how is it different. So similarities, both use DNA polymerase. We need a DNA template. We need nucleotides to make new strands, and both create complementary copies of DNA. The difference is that PCR occurs in a machine, that thermocycler, that cycles through high temperature steps, denaturation, annealing, and extension, and it uses that heat-stable Taq DNA polymerase from Thermus aquaticus bacterium, and that's instead of cell enzymes. So PCR also only amplifies a specific DNA region in vitro, right, outside of the body, not the entire genome inside the cell. For the second question, we have compare RT-PCR and qPCR in terms of their respective purposes. So RT-PCR was reverse transcriptase PCR that converts messenger RNA to complementary DNA, which is DNA without introns, before amplification. And it's used to detect gene expression. We want to see if the gene is on. Are we making those proteins? In qPCR, that was quantitative or real-time PCR. That measures DNA amplification as it happens. So we're going to be using fluorescence to quantify DNA or sometimes RNA levels. And this would be useful in, for example, detecting viral load. In the third question, they ask, in chain termination sequencing, how is the identity of each nucleotide in a sequence determined? So this is the Sanger or chain termination method. This adds specific or special dideoxynucleotides that stop DNA synthesis at certain bases. So each base, that DDNTP, which will be either A, T, G, or C, is tagged with a different fluorescent color. And then we can take those fragments and separate them through that capillary electrophoresis. And then a laser detector will read the colors in order, revealing the DNA sequence. All right, and let's recap 12.2. Explain the use of nucleic acid probes to visualize specific DNA sequences. So a nucleic acid probe is a short, single-stranded DNA or RNA fragment that's labeled with a fluorescent or radioactive tag, and it's going to bind to or hybridize to a complementary DNA sequence in your sample. So when the probe binds, the labeled region becomes visible under detection, and that shows where that gene or where that sequence is located. And the second one, explain the use of gel electrophoresis to separate DNA fragments. So gel electrophoresis separates DNA fragments by size using that electrical current. DNA is negatively charged, so it's going to move down towards the positive electrode, and smaller fragments travel further through those gel pores. After we stain it, we can see the band pattern, which will show the sizes of DNA fragments. For the third one, explain the principle of restriction fragment length polymorphism analysis and its uses. So RFLP involves cutting DNA with restriction enzymes and comparing fragment lengths amongst different samples. Variations in those restriction sites will cause unique fragment patterns for each individual or strain, and we call those DNA fingerprints. This is used in forensics, paternity testing, and also often tracking infectious disease outbreaks, although that's usually done by whole genome sequence these days. For the fourth one, I'm going to save this one for the next slide. I'd like to use a table to compare that. So I'm going to jump to the next one, which is explain the principles and uses of microarray analysis. So microarrays contain thousands of DNA probes attached to that tiny glass slide. Fluorescently labeled complementary DNA from samples will hybridize to these probes. And the color and brightness of each spot tells us which genes are active and to what degree they're active. So we use this to compare gene expression patterns between different conditions. such as healthy tissue versus diseased tissue. In the next one, they're asking us to describe the methods used to separate and visualize protein variants. So we had SDS-Page, for instance. SDS-Page separates proteins by molecular weight after we denature them with SDS detergent. We can then visualize the proteins by staining with Comasi blue or silver stain, and this also can be used to look at protein expression between different samples or disease states. Finally, explain the method and use of PCR and DNA sequencing. So PCR is important in amplifying a specific DNA region using primers, Taq DNA polymerase, and temperature cycling. And this can make millions of DNA copies for studying or testing. We use this in diagnostics, forensics, genetic testing, or research. DNA sequencing, like the Sanger method, determines the exact nucleotide order using those fluorescently labeled ddNTPs. Modern sequencing really uses high throughput next generation sequencing methods to rapidly read the entire genome. And here's our fourth learning objective: compare and contrast southern and northern blots. So southern and northern blots are very similar. They both use gel electrophoresis, membrane transfer, and probes for detection. But the main difference is southern blots, we're looking at DNA. We have a DNA probe to find a specific DNA sequence after gel electrophoresis and transfer to that membrane. In northern blots, we're looking for RNA or messenger RNA. So we're going to use an RNA probe usually, or DNA because it can bind to RNA. And we're trying to measure gene expression. All right, now let's switch gears and look at Chapter 12.3. This section introduces large-scale genomic methods and their applications in biotechnology and medicine. So advanced techniques in sequencing and data analysis have really given rise to the fields of genomics, transcriptomics, and proteomics, and we'll talk about these soon. These fields study genes, RNA, and proteins across entire organisms, and they really form the foundation of personalized medicine. So in the next few slides, we're going to just be looking at vocabulary terms. Our first one is genomics. Genomics examines all of the genes in a genome, and this allows us to compare genomes between different species. We can identify disease by comparing the diseased state versus the normal state. We might even be able to determine resistance genes, so genes that are responsible for allowing a bacterium to be resistant to antibiotics, for instance. And this also supports the development of new vaccines and antibiotics. Transcriptomics reminds me of transcript. Remember, the transcript is the mRNA that we make inside a cell during transcription. So this is called a transcript. So transcriptomics studies all mRNA transcripts made in a cell. And this is helpful because it reveals which genes are active under certain conditions, such as infection compared to unaffected cells, or cancer versus normal cells, or even drug treatment, what happens in a normal state versus after getting the drug treatment. It can also aid in identifying what or when a bacterium or other microbe produces virulence factors inside the host. So earlier we saw genomics and transcriptomics. In regular genomics or transcriptomics, we're usually looking at the genes or the mRNA transcripts in a single species. When you want to look at multiple species at the same time, we add the prefix meta in front of it. So metagenomics or metatranscriptomics, we're looking at multiple species, so usually some kind of community, a bunch of organisms at the same time. So metagenomics applies these same tools to microbial communities directly from their environments. We're not going to culture them in the lab. We study them where they are. So for instance, we could study the genome of some kind of fecal sample and look at all the microbes there, or a soil sample and look at everything in the soil. So this is crucial for studying really the human microbiome as well as the environmental microbes and how diverse they are within their environment. Our next vocab term is pharmacogenomics, also sometimes called toxicogenomics. So pharmacogenomics studies how genetic differences influence drug responses. So it turns out that one drug might have a big effect in one patient, but have no effect in the second patient. So this field, the studies that come out of this field, will allow for personalized treatment plans that really minimize side effects and hopefully improve drug effectiveness. The field of proteomics looks at the full set, all of the proteins in a cell. This is also known as the proteome. Protein profiles, or which proteins are made, really change with disease or environmental stress, so this can help researchers identify different biomarkers for diagnosis or treatment. Biomarkers is short for biological markers, and these are measurable molecules that indicate some kind of biological state, condition, or even disease. And these can reflect normal physiology, like you can have a normal level of these markers, or pathological disease states, such as if you have high troponin levels in your blood, that might indicate heart damage. So biomarkers are measurable indicators of disease, such as having specific proteins that are at higher or lower levels than normal, or different gene expression patterns. So these are used to detect infections, cancer, and track treatment outcomes as well. You might also hear about other omic fields with this suffix. So metabolomics studies all small molecule metabolites in a cell. Glycomics looks at sugar molecules. And lipidomics studies lipids, like lipids in the cell or lipids in the membrane. So altogether, these really omic fields give a complete systems level picture of how cells function and how they respond to changes. And bioinformatics helps us integrate all of these different data types using computational tools. It really helps us organize massive genetic data sets. We can compare genomes, predict protein functions, track disease outbreaks, and many more. And that takes us to the end of part 3. In the next part, we'll look at how these molecular tools are applied to real-world biotechnology and medicine.